This is a story about common prescription drugs, the rhythms of your heart, single cells from hamsters, and the vast universe of big data. Together they formed a grand experiment, an effort to uncover hidden risks and maybe even help save lives. The Tribune's project started with the Food and Drug Administration's Adverse Events Database. It's a huge collection of reports from doctors, pharmacists, drug makers, and consumers who noticed a drug might have caused a problem for a patient. Some drugs are already known to cause a cardiac condition called QT prolongation. It's an abnormality of the electrical system in the heart that can lead to a potentially fatal arrhythmia. Scientists use letters to label the peaks and valleys of a heartbeat. The interval between Q and T represents the period when the heart recharges, refilling with blood. If a drug lengthens the QT interval, even by milliseconds, the heart can start to beat so fast that it doesn't pump any blood. When the brain is deprived of its blood supply, the patient can black out or die. It's sometimes hard to tell from the FDA database which pairs of drugs, taken together, might cause QT problems. There aren't always enough reported cases. The Tribune knew that a data scientist named Nicholas Tantanetti of Columbia University had pioneered a method of finding hidden drug interactions, a way of making the invisible visible. He calls it latent signal detection. The Tribune enlisted another expert who knew all the drugs that caused QT prolongation and their secondary side effects. The team also compiled side effects that were not associated with the disorder. Tantanetti then wrote an algorithm that sifted through all possible drug combinations in the FDA database, looking for this side effect profile. Hundreds of drug combinations, many signals, turned up. But were the signals real? The team checked the suspected pairs against medical records at a New York hospital. Sure enough, some of the combinations didn't turn out to be meaningful. But for 4 pairs of drugs, a significant number of patients who were prescribed those drugs had longer QT intervals. Some of the medications are used to treat infections, high blood pressure, and pain. These results offered real-world evidence that the drugs might be interacting in dangerous ways. But if a biological mechanism could be confirmed in a lab, the findings would be even stronger. More scientists joined the team to carry out cellular testing on one of the pairs, ceftriaxone and lansoprazole. The researchers used suction to pull several hamster ovary cells, which often are used in laboratory research, into tiny wells for testing. Each cell membrane has thousands of Hurg channels, which are conduits for electricity and help coordinate the beating of a heart. When ceftriaxone was applied to the cells, the electric current was unaffected. Same thing with lansoprazole. The drugs alone did not disrupt the Hurg channels. But when the 2 drugs were combined, the electrical current was blocked up to 58%. That provided a biological explanation for why this combination might increase QT intervals. Starting with billions of data points and working down to a few single cells, the team had identified a potentially risky combination of 2 popular drugs. The scientists stressed that these findings are preliminary. Further research and potentially a clinical trial are needed, but the research offers a new model to find more drug interactions, protect patients, and save lives.